I will kick it off with our first question. So the question says, in regards to the viruses that were discussed today, there were no symptoms that were pictures of symptoms that were shown on fruit. Does that mean that all of these viruses are just induced symptoms on leaves? So any of you who spoke today on any of those viruses, are there fruit symptoms that we have that have been observed?
So on the CGMV, showing that those actually showing can show the symptoms on the fruits. So you can have a particular internal symptom that on a flesh, kind of spongy flesh that can be a serious problem.
Anyone else?
Same on the ring spot viruses. It's both of them will cause sometimes symptoms, bumps, kind of on either watermelons or squash. It's very common to see symptoms. Sometimes even fruit malformation is another typical symptom of those viruses.
And we do have an image gallery on the EVCWG website. They do not have pictures of everything on there, but we can also try and see if we can get some fruit symptoms added on there as well. All right, another question. Do federal or state government agencies monitor transplants to prevent the spread of watermelon plant viruses across state lines, especially into Texas, through agriculture oversight, extension services, and pest management? Are samples taken and tested before plants cross those state lines?
Well, since Texas was mentioned, I'll chip in. Not to the best of my knowledge. I think it's a wild, wild west in terms of transplant movement across state lines. It's something that is really very worrisome. I've had conversations with some growers that, tell me, secretly that they, when they are desperate, they get transplants from other states. I've even had a situation where one told me that, well, we opened the box and we saw some whiteflies, which is really very terrible. Yeah, that seems to be a weak link.
Some of those for commercial productions and across the state lines, probably be better, the management for that. But oftentimes it's those nurseries that like a big box stores, those the Walmart's or Home Depot and Lowe's, those actually has a lot of those plants actually cross the state line. You really cannot really have any much of the control of those. Oftentimes, I can see some symptomatic plants and just only walking around those, this just garden centers.
One of the concerns that we have had with many of these viruses, and those that have been mentioned in several of the other webinars that we've had, is that the movement of plant material is only as good as what you're testing for. So often things are not being tested for because they might not even be on someone's radar. It might even be moving in another plant, not even a cucurbit. It could be ornamentals, for example, could harbor some of these viruses. Some of them, for example, the crinkle leaf associated viruses, we don't know that much about them, what they go to. Some of the others, it is good to test. If you see any symptoms in nursery plants, contact the nursery. Those who have nurseries, I know they are very diligent in trying to keep their materials clean, but you can't test for everything. And so it is a concern. And if you can produce within your region, it's always better.
So I would say that probably had some of the county extension, if you had seen some suspect plants, maybe reaching out to the county extension or some of the usually state has some kind of diagnostics labs that can be help you to testing for potential causal agents.
Okay. Let's see. Another question. and our expert on MNSV was not here, but we will see if we can try and answer this question. So it was mentioned that melanocrotic spot virus spreads through seeds as well. Does this mean that if you would use a batch of seeds for a biotest, that the blank or mock plants could also get symptoms without inoculation because it could already be in the seeds?
That can be possible. I think I wrote that down there, but I
Well, in case
and I
anybody said, let's go ahead and repeat it. So,
yeah, so yeah, I think it's possible that that can happen, and that's why I say there that it is important, and especially if you're doing a biotest, is that test your seedlings before, or at least get a set of seedlings and test it with PCR specific for those common viruses to make sure you don't have it, or especially if you're doing with that specific virus, making sure that virus is not there before you do the test. But using certified seed is important. But again, Bill just mentioned that it's very difficult to make sure that the seed is 100% free of viruses. You might have some seed that might be free of that virus, but might have other viruses also. So it's so testing PCR. I think it's important, especially if it's a biotest.
all right, thank you. All right, we have a beet curly top virus question. How widespread is beet curly top in cucurbits or in general in the United States?
Okay, that's an excellent question. And I think the overall answer to the question is the incidence is relatively low and quite sporadic. Okay. So there are going to be many years where there's little or no curly top, but then there's going to be some years where you do get it, and it's very hard to predict. And I'll just point out in 2021 and 2022, in Northern California, this is a little about cucurbits. I've been a professor here 35 years or something, and working on curly top the whole time. We've never had it in the northern part of California in our northern production area. But in 2021 and 2022, we had very hot, dry, windy kind of spring weather conditions. And it blew in leafhoppers, apparently from somewhere, and they spread curly top to our processing tomatoes as well as to the squash and zucchini. Some of those pictures were from 2022. So, and not only that, it was a totally unusual strain, spinach curly top, that we'd never seen before, really, as being certainly not important. It was very rare in California. So, bottom line is, it can fool even people who thought they really knew everything about curly top in California, like me. So but it's very difficult to try to predict bad years. And the good news, as I said, is I think with the removal of sugar beet, certainly from many of these western states that had it, it's really made life hard for the beet leafhopper as well as for curly top virus, because the primary host, both for the insect and the virus, has been taken out of the agroecosystem, and both the virus and the insect had to readapt to these new world crops that aren't anywhere near as good a hosts. So that's the challenge, and it continue. Our growers continue to put yellow sticky cards out. They send them to us. We count the hoppers, we test for the virus, and we can at least tell them whether they need to be worrying or not about the virus. So that's people talk about the challenges with these different diseases. The sporadic and unpredictable nature of curly top is definitely been one of the challenges of this disease. So that's a great question. And again, the disease continues to do things that we haven't predicted.
All right. Let's see. We have some watermelon crinkle leaf associated virus questions now. So, Femi, you are now in the hot seat. The first one, What symptoms have been associated with watermelon crinkle leaf associated virus 1 and 2 and other cucurbit crops? And were there any unusual symptoms associated with those infections?
So again, there are just very few reports of these two viruses, fairly crinkle leaf associated virus 1 in other cucurbits. As a matter of fact, in our own study, we exclusively found them in watermelon, with the exception of 1 or 2 samples of smell melon and a sample of pigweed that tested positive for the virus. But for instance, the first report of the presence of crinkle leaf associated virus 1 in zucchini squash from Florida documented, they showed, symptoms of some yellowing and crinkling. What is not clear from that first report was if they excluded, the presence of other viruses in that particular plant, because again, in our own experience in Texas, we've not really found either of those two viruses as single infection. So I will be very careful interpreting symptoms. I know there's a lot of reports from elsewhere, from Georgia, but also from outside Georgia, that have really shown, that with crinkle leaf associated virus 1, you can, really associate it with, some kind of leaf deformation, and in some cases even some kind of discoloration. So I'll leave it at that, and I'll say, if you look at any suspect plant and you find a positive for either of these two viruses, please test for other viruses to just see what the viral dynamics is in that plant.
All right, and let's see. Sudeep, I did give you speaking privileges if you have any comments that you want to add into some of these questions about watermelon crinkle leaf associated virus. The next question is about the seed transmission of these viruses. Do the published experiments demonstrate transmission to the adult plants as well, with disease symptoms developing in those plants? It says, based on the slide, it seemed that the experiment only demonstrated that the viruses were seed-borne, detected on the seed tissues and radicle of the germinated plants. So we're interested in knowing, did the developing plants exhibit symptoms?
That's a very good question. So going back to that slide, I showed a picture of localization of the virus in different parts of the seed, but also I showed those bar diagrams that showed the seed transmission rate. So what happened? So first and foremost, that study was done initially to just generate antibody for ELISA detection, because these are two viruses that are listed on the EPO list, and they wanted to generate antibodies to enable, rapid diagnosis, because up until then, everybody has been relying on just molecular diagnosis. And it was in the process of trying to test the antibodies that they, found, some evidence of this seed transmission. Of course, it's been suspected all along. And what I showed in that bar diagram are plants, are seedlings, actually, seedlings up to a couple of weeks, old seedlings that were produced from seed lots, including seed lots that dates back to about 8 years, before this study was conducted. And what you see in those bar diagrams are, the rate of detection of either WCLAV1 or 2 in those different cultivars based on immuno detection performed on the seedlings generated from those seeds. There is no information in that publication of symptoms observed on those plants. I suspect this is something that they are probably still, working on. Maybe the paper was just focused on being able to say, well, we've generated this antibody and it could be used, for, in epidemiological studies and things like that. So I can't speak to whether there are symptoms, but what I can say is that those plants, are systemically infected. So the other part of, that slide is just basically them asking the question, is this just a contaminant of the seed coat, or does it go to different parts of the seed? And the answer is, it's not just on the seed coat. it goes into the cotyledon, to the radicle and different patterns. So, disinfection may really not be something that really will be very effective to manage any contaminated seed lot.
All right.
Can I make one comment here?
Sure.
I'm very happy to hear that you're bringing this point up about whether the symptoms develop in those seedlings, okay? Because, it's very important to show that and to show that it's kind of a natural infection that develops. And a number of these studies, recent studies arguing seed transmission, and showing that, yeah, it's in seedlings, but they don't show the symptoms. And I call these, atypical or whatever, because there's no evidence, these can be silenced genomes of the virus, okay? The plant in its reproductive tissue strongly silences viruses and things. So these can be non-viable viral genomes that are being tested in the progeny seedlings, and it doesn't prove that they're biologically active. And if they don't induce the symptoms in those progeny seedlings, that's what you typically see. Kai mentioned the 3 seed-transmitted viruses that, have been clearly demonstrated in this way to be, seed transmissible. And of course, the progeny show very strong symptoms. So we have to be careful, when we're making these conclusions about the biology. Now, on the flip side, on some of these so-called seed-transmitted viruses, they don't fit the epidemiology of the disease either. Tomato yellow leaf curl is a classic example. Okay. However, in this case, seed transmission does seem to fit, the epidemiology of the disease, how it's spreading so quickly. Okay. So, we need to consider all of those factors, I think, before we declare true seed transmission. And I think that would be to the benefit of everybody. So thank you for being able to, add that to this.
All right. Another watermelon crinkle leaf associated virus question. Are we aware of any research that has been conducted on the effects of the virus on yield and soluble solids? So again, this is for a watermelon crinkle leaf associated virus.
As far as I can tell, the answer is no. To the best of my knowledge, I don't know if Sudeep has anything to add to that, but I haven't seen any information, in that direction. So it's one of the things I mentioned in my slide that we still do not know, what their impact is, whether by themselves or in mixed infection with other viruses, in terms of, impact on yield as well as quality.
Sudeep, did you have any comments about any of them?
No, I don't have any comment. I agree with Femi on this. Yeah.
Okay, and then we do also have a comment. Again, this was addressing the fruit symptoms caused by watermelon crinkle leaf associated virus 2. They're saying in Italy, they observed necrotic rings on fruits from plants that are infected with watermelon crinkle leaf associated virus 2. Along with watermelon mosaic virus. So, but I think you all have both said that you don't really know, can't comment on what is definitive in terms of fruit symptoms, based on soil infections. All right, let's see. Our next question, looks like we are now shifting to CGMMV. So we did have a question asking for Dr. Ling to share the literature about how TSP is effective in disinfecting seeds of CGMMV, and I believe he shared that in the comment in the chat box. So we have another question. Why and what is the chemical and heat treated seeds are still testing positive?
Very good question. So what happened is those treated seeds that you are testing, the current testing that you're using ELISA or PCR, those are can detecting both living virus or dead virus, if it's like infectious or non-infectious. So still, after treatment, you still can detecting those. So to determine if those viruses are still can be infectious, then bioassay is necessary. So it had to be through this mechanical inoculation onto the indicator plants, like some of the cucurbits, particularly cucumber, very easy to get symptoms on that. And then you can actually, so you won't go back to using the ELISA PCR test again. Otherwise, it's overestimating this on the risk that on those treated seeds.
All right. Our next question is, do you have evidence, and this covers not just CGMMV, but do you have evidence of the percentage of incidents or losses of yield of watermelon crinkle leaf associated virus, CGMMV, or melon necrotic spot virus in watermelon, squash, melon, or cucumber in the United States? So we already addressed this for watermelon crinkle leaf associated virus. Is there any documentation for some of these other viruses in terms of their incidence or yield loss in the US?
I would say on the CGMMV, probably in California had more experiences on that, but oftentimes it's good thing is that the CGMMV, even in the United States, even though that was showing all the map, all red there, but really it's all localized into California. Currently not being officially reported to other states or widespread in other states. So that's kind of good news on that. But how much the yield lost, I think it's more of the impact about actually it had the rotating to other crops for several years, and then but I don't have any particular data for that. I'm not sure if Bob has any information on California situation.
I can just say that we test a lot of cucurbit samples for virus symptoms every year in California through our virus surveillance system for vegetables. And we have not encountered a CGMMV in any of our samples. And as Kai mentions, I think the outbreaks that were identified through our Department of Agriculture, Dr. Bryce Falk from our department was working on it too, have been very limited, and they've mapped them out and they've followed them carefully and quarantined fields, and they've seemed to have done a very effective job of minimizing it. And the other issue is the Hmong community. Some of these communities, grow traditional cucurbits and things. And, they sometimes bring in seeds and stuff, from other locations. And this has been a tricky area to handle. Okay, so it may still be there in those type of in those gardens and things, but it hasn't jumped into our commercial production, and we don't run into it. Same with melon necrotic spot virus. It was first found in California, as it was mentioned in 1935, but that's one we haven't encountered in my career very much at all. Okay, so I don't think it's really much of a factor, nor tomato ring spot and those Nepo viruses too. So we're always looking. But fortunately, I think the CGMMV situation in California has been pretty well taken care of so far. And that's kind of where it is today. Certainly, it's not causing any losses. Our growers are not even, really worried about it. And we, so fortunately, that's kind of how that's developed.
And I will
So most
say,
of,
go ahead, Kai.
yeah, I would say that most of those the seaborne or mechanically transmitted tobacco viruses, since I'm working also on the tomato brown Lugo fruit virus, is that the most of those are in more like greenhouses, the more mechanical transmission. So if you're getting one plant get infected, then you can actually do this mechanical transmission, really causing a lot of trouble in that greenhouse production or in the food piles that you actually kind of the leaving and more kind of manual, pretty much because it's not vector transmission, so it's actually the man transmitting as a kind of becoming the vector, the handling of the plants and that kind of thing. So that's usually the problem there.
Yeah,
I was saying, I do want to point out that I included the link to our cucurbit viruses in the US cheat sheet. WGMMV and CGMMV are both listed on there, but they are listed as being transient. And so I do want to reiterate that both of those are considered to be under official control in the US at this time, but they were significant threats. And so we did want to cover them. And we also have.
Just to add very briefly, CGMMV is still regulated. So in terms of impact, that can be a very big problem for the entire industry. Just wanted to add that.
And we do have Tong Yen Tian from CDFA, if I am correct on that. Did you have any comments about the CGMMV in California?
Good.
Hi, yes, can you hear me?
Yes,
Yes.
we can hear
Yeah.
you.
Yes.
Yeah, so in terms of virus the damage, I think we in California we have never allow that situation to be evaluated because once the virus has ever been detected, it be immediately going to quarantine. So everything would be if you say direct damages will be the loss of crop that is due to the quarantine. So the yield and everything that what being observed in other countries no, we do not directly. That means currently we are unable to assess that just because the regulatory situation. So that is on the CGMV, and I think this bar still the seed production, the seed lot is still being tested by the company, the seed producer themselves, and then they have a self-reporting system, and every time it's found, it will go through some regulatory process. And another comment on the watermelon green model mosaic virus. I think my personal opinion is that the name is rather unfortunate, and it's mostly associated with Asian melons, Asian cucurbit. So I think we initially found it in bitter melon. So but you carry that name, it doesn't seem to be the primary watermelon virus. So that's my what I have here.
Thank you.
I hope this helps.
Yes, we appreciate the input. All
Yes,
right.
Andy, I think it's important to say, in short, California managed this really well from the very beginning, and it's never really been a problem here. So thank you, Tong Yan, for clarifying how that's played out. And Tong Yan works with the California Department of Food and Agriculture and has been on the front lines of virus detection in the state for many years.
All right. Another question related to CGMMV. What are the major gaps in understanding CGMMV host interactions? And then are there any ongoing global surveillance or breeding programs targeting CGMMV?
So it's kind of really challenging on the looking for the source of resistance, and I think we actually exhaust our watermelon almost over 1,000 sections that what we actually screening, particularly on watermelon. We didn't really see any good source of resistance. As I mentioned, just kind of more tolerance. It was really challenging to even working on for genetics study on that, and so. And I don't really see any. There's some efforts in the seed companies, but again, it's really quite minimal work that currently have. But maybe they will be looking into some of the biotechnology. So we'll see what what we could be happening, like gene editing or CRISPR type of technology could be developed. But currently, there's not genetic resistance, just not really there.
All right, let's see. We had a couple of questions related to the information you presented, Kai, on the seed treatments. Looking about the efficacy of the seed treatments with dry heat and TSP. And then also someone noticed that Physan 20 was not on the list of chemicals that were tested against CGMMB. And they would like you to highlight why that may be the case, or if perhaps that chemical is represented by another product formulation.
Yeah, we were screening these kind of just selecting some of the common ones that use in the industry, but maybe that was one that we did not include at that screening. At that moment, we were looking at both tomato brown rugose and CGMMV together. But yeah, those I mean, there's a lot of companies, the chemical company wanted to include in those, but we had to kind of had to selecting certain ones. Yeah, that particular Pfizer was not included in that test. But those TSP, yeah, it's really commonly people are using that for seed treatment, and we're looking at some of the newer ones like Voracid, which is very effective. I mean, actually, the most tomato greenhouse growers are shifting to use those Voracids rather than using the Clorox, because the Clorox, particularly in the greenhouse, that the metal can be corrosive to equipment and facility like greenhouses structure. And so they actually kind of more shifting to Voracid. So if you initially was used that for the veterinarians, but then I actually, through our screening, working with the manufacturing and distributors in the US to actually getting these the EPA to changing the label to allowing that using in the horticulture crops. So now I think it's already available there that you can try to use those Voracids. I think that seem to be the economical and very efficient for tobacco Voracid. Not just tobacco, I think there's other Voracids can because it really broad the effect on not just only these tobacco Voracids, but other Voracids could be too.
All right. And then we have another beet curly top virus question. What is the main criterion used to classify beet curly top virus strains?
Okay, that's a great question. Where the strains are identified using a pairwise DNA sequence analysis. Okay, so they use the complete genome sequences of the curly top virus isolates, and then by using pairwise comparisons, Arvid Varsani and his group in 2014 published a comprehensive analysis of all the BCTV sequences out there and came up with 11 strains of the virus. So before that, they had named them species. There was a beet mild curly top, a beet severe curly top, and just a beet curly top. But then when Varsani did this analysis, they identified, in fact, that these were not truly species. Their cutoff values were much lower for species, 77%, whereas strains were 94%. Or less. So now we're dealing with these 11 strains. They're identified based on nucleic acid sequences. And in our pretty much extensive surveys of curly top in the Western US these days, these 11 strains are the ones that continue to be identified. So in other words, we may have an idea of pretty much the genetic diversity out there. I don't, certainly new ones can easily come up, but right now we seem to come up with these 11 strains, and in California, there's about 5 of them that are particularly prevalent. They're more of a problem in processing tomatoes. So that's how that's determined. So as I said, we have this PCR test that differentiates the so-called mild type and the severe type strains. And that's very useful because many of the severe type sugar beet strains, as I said, are no longer found, or they're much less common. And now these mild type strains have emerged, and they fit in a very different kind of lineage. So there were two original lineages after sugar beets were introduced. And these mild type strains have, as I said, emerged now as the most common ones. And they can be identified by DNA sequencing, but we can at least differentiate the mild type ones with this initial PCR test that doesn't require us having to do the sequencing. So yeah, it's not a totally simple situation identifying which exact strain you have. So I hope that makes sense.
All right. And I believe we have just two more questions. One of them is a more general question. Are there any cross protection measures tried for these viruses? Or, in other words, are any mild strains or attenuated virus strains inoculated in experiments against cucurbit viruses?
I would say that very early on the zucchini yellow mosaic virus has been demonstrating to give good cross protection, but I don't think anything that actually going into currently for like a commercial production to managing that disease. And these other viruses, so far, it's not really have this cross protection that I aware of.
Anyone else want to address that?
Yeah, well, I'll make one quick comment on it. Like Kai said, zucchini yellow, and I think in Taiwan, they've done a lot of cross protect, not only with papaya ring spot, but I think into cucurbits too. Shadong Ye, and so remember the first transgenic plants, okay, from Asgrow Seed Company targeted cucurbit infecting podaviruses, ZYMV, WMV, and PRSV. And the idea was based on cross protection, right? That there were some mild type strains. And so they made coprotein transgenic plants. And I'll tell you, the ones for ZYMV were great. They were so resistant to Z. We used them in our introduction to plant pathology class to show the kids how that transgenic strategy actually does work. So certainly, the cross protection that was found traditionally, led to the idea of the genetic engineering and taking advantage of RNAi, because of course it turned out to be RNAi, not actually the capsid protein itself conferring the resistance, right? Turned out to be gene silencing. So really, I mean, somebody mentioned, I think Kai brought up genetic engineering, and surely that is one way in the future, that we should be optimistic that strategies are out there that could be developed, but you have to get over the hurdle, of course, GMOs and all that type of stuff that's still out there, but maybe, becoming a little more accepting of some of these technologies. So I think there's a lot of promise in there. But again, we heard a lot of promise about the cloned R genes and all that type of stuff, and that was going to change plant breeding and everything. And of course, it didn't. We're still using the traditional breeding and stuff really to protect our crops and IPM strategy. So we should be careful not to throw all our eggs in one basket of CRISPR, because we heard the same thing 30 years ago about the R genes, okay? So you have to be a little careful. CRISPR is a powerful technology, no question about it, but it has a lot of, issues and, non-target effects, for example. So, you can't be telling our growers, hey, CRISPR is going to solve your problems, because I think that wouldn't be a accurate statement. So But you know, really, and you know, talking about those Nepo viruses, you know, we had this virus that showed up. It's not a Nepo virus, but it's an ILAR virus that jumped into tomato. Okay, it caused like spotted wilt type symptoms. It's called tomato necrotic spot, but the tomatoes recovered from it. Okay? And then it never became a problem. So we were able to tell our growers, hey, this is one virus you don't have to worry about. Okay, the plants recover from it. You can't even tell it was even there if you go back in July. So it's a powerful, you know, natural immunity that plants have, and if you can, you know, harness it the right way, it would be great. And let me plug the beet curly top, the beet leafhopper. If we can find, you know, designer peptides that you could spray on the leafhoppers that get taken up and, you know, muck up one of their critical, you know, metabolic pathways, it would be a very nice designer type insecticide rather than, you know, other non-specific insecticides like malathion that's been used by the beet curly top control program here. So, and, you know, even though that's not a, you know, really highly toxic insecticide, it has an on-target effect. So anyway, thank you for I hope these new methods like CRISPR are going to solve the lack of resistance.
Okay, well, that addresses all of the questions we had pertaining specifically to the viruses that were presented on today. But Bob, we started out with you, and I guess we'll finish with another question for you. I do want to thank you all for joining us. And this question, I will let Bob give a short answer to this question,
All right.
but he did do a webinar on this, and I will include that link in the chat. So today's webinar, it focused on seed transmitted viruses. That was one of the foci. So the question is there any evidence of seed transmission of begomoviruses from the New World? We are obtaining somewhat mixed results for seed transmission of Old World begomos. Some are reported as transmissible and others are not. What are your reviews, and should we be testing seeds for begomos? So first, you may want to explain what Old World and New World is for those who may not be aware of that. And then give us your thoughts. And I will include the link to his previous webinar, our first EVCWG webinar, that is available in its entirety in the chat while he answers that question.
Well, you weren't going to let me get away here easily. Okay, I'll make it quick. So, like I told you before, you know, one thing about Gemini viruses, I have to tell you this, they're ancient viruses, okay? They evolved from single-stranded DNA plasmids that were in prokaryotic organisms or microbes way back in early evolution. I'd say, you know, billions of years ago, okay? You know, but certainly hundreds of millions of years ago. So why am I telling you that? Well, there was a huge diversity of these viruses that established interactions with primitive plants. What that meant was when the continents spread, Gemini viruses were all over the place, okay? So they were jumping out of plants and infecting different crops that were planted there, causing these viruses to emerge. What happened was there were whitefly transmitted viruses on this Pangea, you know, that emerged to whitefly. And when the continents divided, you got old world type ones who evolved independently after continental drift, and new world type ones that evolved in the new world, okay? And the most notable differences are many of the old world ones are monopartite. They don't have a bipartite genome of 2 DNAs, and they associate with a satellite DNA, which the virus acquired from some unknown source, okay? That suppresses gene silencing, actually. In the New World, most of them turned out to be bipartite. They have a DNA A component and a DNA B component, okay? And both of them are required for infection. But like Gemini virus always do, they can never be predicted. And now we have monopartite New World begomoviruses that we've identified too, okay? And there are some old world bipartite ones. But in general, you could say the old world ones, you know, are pretty different than the new world ones. Now, to the situation of seed transmission. Okay, long ago, when they found these diseases, they looked to see if there was any evidence of seed transmission doing the usual grow out type test. And they found no evidence of seed transmission of curly top, maize streak, any of the butylon mosaic, a bunch of these things. Okay. And so then when we got the molecular tests, we've started to revisit the situation. Okay. And what we're finding is that the Gemini Pagomoviruses, they spread, they're phloem-limited viruses, and when they get up into the reproductive system, they certainly infect seed-associated tissues. The seed coat for sure, okay, and then they can even move into the seed and be detected in the embryo and other seed-associated tissues. Okay, so they're definitely getting in there. When you do a PCR test on seeds, you can detect it. Now, we did a beautiful experiment back in the day when we labeled one of these viruses, BDMV, with the green fluorescent protein because they didn't need the capsid protein. We could put the GFP in. We followed it moving up the plant into the flowers, which it did, into the pods. It was beautiful, but it was limited to the seed coat. It never moved out of the seed coat. It stayed in there, but it was definitely in the seeds. It was definitely in the flowers. Okay. So, and this virus was never, BDMV was never reported to be seedborne. So now you're seeing a situation where you see this detection in these reproductive organs. The question is, does it become productive seed transmitted virus? Okay. So there comes back to this issue of, yes, when seeds are taken from these flowers, okay, and planted out in a grow out test, they either don't show any symptoms or, they're this, atypical type kind of detection only by PCR, which is not the natural way these things, well, if they're going to be seed transmitted and be biologically relevant, that isn't going to do it for the virus. So a number of people said, okay, this doesn't make sense with the biology of the disease, like tomato yellow leaf curl. And then Perez Padilla in Spain did a very nice study where they did this to look at more natural-based potential for seed transmission of TY by collecting seeds from naturally infected fruits, planting them, found no evidence of seed transmission. Same with TY-LCV in Sardinia by the Italian group, okay? And so the emerging story is that, this is probably not biologically relevant. We've always said, then why don't we see TY-LCV popping up all over the world where they plant this hybrid seed that was produced in places with TY? We should be seeing TY showing up in California then, theoretically, which we never have, okay? So this is the issue, okay? It doesn't fit. And also, it was the same thing was done by Kai and the folks in, I think you were a part of that paper, Kai, where they looked at sweet potato leaf curl virus, which was the first one of these PCR seed transmission reports. And they too, when you did the grow out and you did it the right biological way, failed to find evidence of seed transmission, okay? Correct me if I'm wrong, Kai. So my opinion is that we have not presented sufficient convincing evidence that these are truly seed transmissible, nor is it consistent with our extensive knowledge of the biology of the diseases, making it, in my opinion, premature to make decisions about, having to test all seeds for Pagomoviruses. I mean, that's a big ask of our seed companies. And I think we should be very careful with the science, before we, would encourage that to be done. So that's my personal opinion on this. I can assure you, I've thought about it extensively. I've read many of these papers, and, that's my opinion. I know Bill is also working on it. He may have something to say. So thank you. Thank you.
We're way over our time.
Okay.
Thank you, everybody, for staying. Thank you, Bob, for that explanation. And I did put the link to Bob's webinar where he did go more in depth into the things that he was mentioning. So feel free to go back and visit that. Again, there will be a short survey after. We appreciate your feedback. We hope to have more webinars in the future. And so we do look at the input that you give us for what you would like to hear about. So thank you all very much. Have a great day.
Thanks, everybody.
And thank you to all of our speakers as well and panelists.
Thank you. Thank you.